Namaste students, खुश हो बेटियां. I, JV and Dr. Vaishnavi, Assistant Professor Jyoti Vidyapeeth, Women's University, Jaipur. In today's session, we are going to study about the mitosis permanent slide. Okay. So here we have a different phases. In the screen, you can see the different onion tips showing different region of mitosis. Okay. This is the LS of the section which shows the metaphase, anaphase here, interphase, and two total nuclei, and the telophase and the prophase. When we are going to see a specific slide in a particular phase like interphase, here you can see the cell wall and the cell membrane and the nuclear membrane with the condensed nucleus and the chromatin network can be found. So in this stage, prior to the actual mitotic cycle, the cell appears to be the inactive and in resting stage, but is metabolically the most active DNA replication occurs during this phase. The nuclear membrane and the nucleolus are very distinct. So the chromosomes are in the form of chromatin and the network, and the individual chromosomes cannot be seen separately. So here the chromosomes appears a double stranded that is made of two chromatids. The next stage is the early prophase. In the early prophase, the first stage of the mitosis can be observed under a microscope where the nuclear membrane appears distinct. The nucleolus also seen clearly and the nucleosomes become spoiled and shortened and more distinct. In the late prophase, the nuclear membrane and the nucleolus have a partially or a completely disappeared, and each chromosomes begin to show the chromatin, the primary construction and the secondary construction of the centromere. The equatorial region appears clearly in the center of the cell. The chromosome begins to move and gather near the equatorial plane, and the chromosomes are condensed and thus short and thick. The spindle fibers also begin to appear. In the metaphase, the nuclear membrane and the nucleus are absent, having disappeared. All centromere of the chromosomes are appear reached in equatorial plate and each is attached to the spindle fiber. The centrioles are absent and hence astrosis is not found in the plant cell. This type of mitosis is known as the anastral mitosis, where the spindles are made up of fibers only and the absence of the centromere indicates that it is a plant cell. The centromere and the metaphase are very distinct, and thus the number of the morphology of the chromosomes is studied in this stage. Each chromosome shows two chromatic chromosomes or a primary construction or a euchromatic and or a heterochromatic region or a centromere, etc. The fifth phase is the anaphase. In this phase, the complete in which a very small period of the time or a centromere of each chromosome gets split into two. The chromosomes also get divided into the two chromatid and each chromatid now bear one centromere each, and the chromosomes become shortened and thickened. Separate chromosomes are now pulled towards the opposite poles and due to the contraction of the spindle fiber. During the movement, each chromosome shows a characteristic shapes which depend on the position of the centromere. So here you can see in the screen, this is the cell showing the early prophase and showing the metaphase. In the prophase, the cell wall or the nucleus is condensed and the chromosomes you can find it here. And in the metaphase, the chromosomes are on the equatorial plane. Okay? And the poles are formed with the spindle fibers on this. Next is the cell showing the late prophase. In the late prophase, the nuclear membrane and the nucleus disappears here, and the cell centromere can be visible, and the spindle fibers are formed. In the anaphase, these spindle fibers are moving towards the poles. You can see here the centromeres are formed and the spindle fibers or the chromosomes movement and the aggregation near the pole starts. Next, the cytokinesis takes place where the cell divides into two daughter cells, and the movement next to that is the telophase. In the telophase, the chromosomes are present in both the poles of the parent cells, and the chromosomes increase in length and become thread-like. All the chromosomes together forms the chromatin, and the network and their individuality is now lost. The group of the chromatin network at each are surrounded by the nuclear membrane. The nucleus is present. The two fully formed nuclei, uh, one at each poles, are present in the parent cell. The spindle fibers are absent. The cytokinin in this stage of the cytoplasm divide into two. It results into the formation of the two daughter cells. The division of the cytoplasm is due to the formation of the cell plate and the equatorial region. The cell plate form begins in the center of the cells and gradually progress towards the periphery. So this results in the formation of the two daughter cells. Organelles can also present. So the number of the chromosomes at each daughter cell is equal to the number